Hey, thank you, Carl. So I'm going to tell you about AI in structural biology and the roots of success. And before I even get going, Carl knows this. I am not a computer scientist. I only know a little bit about AI, but I do know something enough to know why there was such success in our field, and that's what I'm going to talk about. So this is a paper written by a colleague of mine with whom I collaborate, named Joel Gershenfeld at Brandeis, and it's a good title, Garbage In, Garbage Out. And that's pretty much what I'm going to talk about here. And Joel and his colleagues point out the importance of data curation in having success in AI. And the example I'm going to show you, and I'll show you how, we had extensive data curation, and I'll explain what I mean by that. So, sorry. So in the journal, the New York Times, in 2020, there was an article saying that there was a big breakthrough in AI that would accelerate drug discovery. And specifically what this was that they were able to take sequences of proteins and predict the structures, which is one of the grand challenges in the last century and this century, and they were successful. And I'm going to tell you a little bit about that and why they were successful. And so to start, to be fair, I think I need to tell you a little bit about what proteins are at a very sort of high level, let's say. But protein
Hey, y'all, we can't hear you. ISI, I think you're muted. Got it.
So what happened?
The room muted. Did we touch the VTC by any chance?
Yeah, I don't know.
Can you hear me now?
Yes, they say.
You can.
Remote people.
No, they're texting it,
Oh,
yes.
they can. Okay, good. So basically, we think we know that structure determines function. We think we know there are approximately 20,000 unique proteins in humans. And human proteins are very similar to proteins from other species. I mean, really similar. So,
Can we ask weird questions? Wait at the end.
what?
So can we ask weird questions?
It's up to you how you want to do it.
I'm just curious. So the 20,000, I know that proteins, they have different ways of being read. So you can put in fragments. Just
Yeah.
with a protein, it can be read humans in different ways. You can splice different parts out. So is that the 20,000 or the 20,000?
20,000, the estimate is that there are 20,000 unique proteins, meaning whole proteins, okay? The way in which we know, we don't always know the structure of a full protein. We might know only the structure of part of a protein, okay? So I can explain that too.
Yeah, okay.
Okay, so here's just a diagram showing the diversity of protein structures in the world, and it's done as according to function. And so there are structural proteins. On the far right are protein-nucleic acid complexes. Those round balls are viruses, and so on. So there are a lot of different proteins. This particular picture was made by a colleague named David Gutzel at Scripps, who's very good at depicting the structures. In terms of the protein chemistry, they're polymers consisting of defined sequences of amino acids, and they're connected as shown on the right. And they're connected via something called peptide bonds, which are shown over here. So you have basically two degrees of freedom. Everything else is flat. And so you basically have 2 degrees of freedom per residue. And the preferred areas, the preferred conformations are shown in this plot called the Ramachandran plot, which tells you the kind of allowed areas of conformation. And so that's what a protein is in a very pretty high level. There are levels of structure that we talk about. There's secondary structure. For example, on the left is the alpha helix, which was first predicted by Linus Pauling in 1948. And it turns out this is a very important secondary structural element. There's also something called the beta sheet. And those are two primary sort of local structures in proteins. The proteins then fold into what's called a tertiary structure. And fortunately, the first such structure that was done really consisted of basically mostly alpha helices, which was lucky in being able to determine that structure. And then there's something called quaternary structure, which is how things pack together. So for example, hemoglobin has 4 subunits packed together, and hemoglobin is what carries oxygen in your blood. So, so that's basically what a protein is. It's a polymer. Each residue has 2 degrees of freedom. There are allowed regions and disallowed regions. The first protein structures were determined in the 50s and 60s, mostly in England. On the left, the first one was myoglobin, and then on the right is hemoglobin, then something called lysozyme and ribonuclease were the first structures, and they were a really big deal because nobody had any idea what the 3-dimensional shape of a protein would be like, and so this caused a great deal of excitement. So until the 1980s, the main method to determine the structure of proteins was using something called X-ray crystallography. And the method consists of growing crystals of proteins. So the crystals are pretty much the same as a crystal of quartz, except it has a lot more water and a little more delicate than quartz, but principles are the same. The experiment consists of taking the crystals and putting them in front of a X-ray beam and then getting a diffraction photograph, which is an indirect image of the protein. So in order to, so you get all these spots shown in the third panel, and those are called X-ray intensities. And then using a whole different bunch of methods, which we're not going to talk about today, you can take those spots and figure out what gave those spots. Well, I will say that every spot contains the positions of every atom in the molecule. So every single spot contains information about every single atom. So the methods for deconvoluting this are quite sophisticated and have won a couple of prizes and lots of recognition. This was really important. And then you get a model after you solve what's called the phase problem, and you get the structures shown here. This happens to be a protein-nucleic acid complex, but you can look at every single crystal structure is pretty much determined the same way from the beginning of time. It's just right now we have so much better computing and so much better technology that it doesn't take a long time. So it used to be, it took 35 years, now it can take a day. And so that's what's involved in doing a structure. At the same time that this was going on with determining structures, there were people trying to understand protein folding, was being investigated both experimentally, and on the left is someone called Chris Anderson. And Chris was able to demonstrate through various kinds of experiments that the sequence of a protein, the sequence of amino acids in the protein, determined the structure. How that's done, he didn't know, but he was able to show that that was the case. Cyrus Leventhal was a biologist who was very interested in computational approaches to protein folding. And he was one of the early people to begin looking at protein folding and trying to understand it. So the protein folding challenge was, how does the sequence of proteins encode for the structure? How does that actually happen? And how do proteins fold so quickly? Because you've got a huge number of things in this polymer, and each one has 2 degrees of freedom. How does it happen so quickly in the body? And that is really what protein folding is, not what the New York Times says it is, okay? So that is the real challenge. In order to understand how proteins fold, you need to know the sequence, which is the sequence of amino acids in the polymer, and you need to know the structures. Back in the day, in the 60s, exchanging data was a laborious process involving sending punch cards and boxes or tape via snail mail. And so if you wanted to look at a structure, there was no easy way to do it. And to ask somebody in England to pack up 6 boxes of cards and send them was ridiculous. So we needed to have a central repository. And that idea came in the 1960s because of all this work that I just described. Being that it was the 1960s and I was part of that generation, we thought we had to be very active, and we had petitions. We didn't have protests, but almost. And we had a petition saying there has to be a protein data bank. Why, who we thought was going to listen to us is not clear, but we thought that we had to do that. What happened then is that there was a meeting in Cold Spring Harbor in New York. It was the first meeting in 1971 that described the structures of proteins as they existed at that point. There weren't too many structures. A bunch of us crashed the meeting, because we weren't invited, but we knew that it was going to be an important meeting. We went to the bar after the session was over, and we approached someone named Walter Hamilton, who was at Brookhaven, and we said, we need a protein data bank. And he said, okay. And that's how it began. So he flew over to England because he understood that it had to be a global enterprise, and set up the PDB. He set it up at Brookhaven. Edgar Meyer, who was a visitor at Brookhaven, and I collaborated on the technology. And we formed something called CRISNet, which would allow data to come in over DARPANet. And the way we did that was there was a key punch, a paper tape thing in the animal colony where I worked, and we were sending data back and forth in that way over DARPANet. And so that was the beginning of the PDB. It was based on this passion. It was based on huge amount of technology and the understanding that there was some kind of gold in those structures. So what does the PDB contain? It has the atomic details of the molecules, the name of the atom, the residue number, coordinates, something called temperature factors, which tell you how the atoms are waving around, and occupancy factors. There's the primary structures in there, that's the sequence, the secondary features, such as helix and sheet, experimental details, primary data, and citations. There's a lot more in there, but this is the summary of what's in a PDB file. Okay, so we had the PDB, but who says anybody should use it? Who cares? Well, several people cared, and they said that it had to be mandatory, and that we had to have mandatory deposition for publication, which was very clever because scientists rely on having publication. And if they can't get something published without having the data in the PDB, that's a big problem. And so again, a bunch of committees were set up, lots of activism at that period. It took 7 years to figure out exactly what we mean by mandatory deposition and what data actually should go in, because you don't want to put in a whole bunch of garbage. You want to really have very well-defined data. And so the experts in the field got together, argued, until they came up with a plan, and that was published in 1989. And that then essentially meant that all protein structures eventually, it took a while, would be in the PDB. So we would have that central repository that we so much needed. The other issue, which was very important, were data standards. The way in which the data was structured initially was based on the 80-column format of a punch card, and it was an implicit format. So you didn't really know if one of you guys came in and tried to figure out what was in those files, it would be very hard. And so we decided to have this new kind of format. It's called MMSIF. And I'll show you a little bit about what that looks like. That was done in the early 90s, and it took until 2011 for people to finally say, okay, the PDB was using it, but nobody wanted to use it because it's too complicated, they said. And it took a long time to get acceptance. This is what part of a format looks like. And it's a very simple syntax. Every day it's an explicit definitions. There's a DDL, a dictionary data definition language, and then there is the dictionary with all the terms, and I think now we have about 7,000 terms. I think we did about 4,000 terms when we started. A PDB file doesn't have that many. It only has about 400, but it turns out that our decision to try to define everything was the correct one because things began to expand. And so this became the format. It meant that anybody could read it. I don't have enough time to go through this, but it's basically a whole bunch of tables and a very clear hierarchy, and it allows you to make, if you wish, a relational database with the data. The business of having global cooperation was also important because there was always a danger that different people would start making different PDBs, and then you would never know which is the correct data. So we have an agreement about data processing and annotation, which is what I'm going to talk about. And so the agreement is that we would all use the same way of data processing and annotation, but we could compete on websites and services, which is fine. And again, this first slide that I showed you from Joel's work, Joel Gershenfeld, is that if you want something to work in a community, you have to define what you're going to cooperate on and what you're going to compete, because we're human and many of us need to compete in order to feel we're doing something important. In 2008, even though the PDB would accept the primary data that's underneath those coordinates, it did not require the primary data, and so it was decided that we needed to have that, and that is really important when I talk to you about validation and structure annotation. And so that happened in 2008. Again, you'll see there's a theme here over and over again that you had to get the community to buy into all these things. And so it took a long time, but it's extremely solid. X-ray was the dominant method, but then in the 1980s, nuclear magnetic resonance became a method for structure determination, and that earned Kurt Ruettig a Nobel Prize. And in this case, you could determine the structure in solution, and that offers many advantages because not everything can crystallize. And so PDB began to accept NMR type structures. More recently, in the 90s, the method of electron microscopy became popular. And in that case, what you do is you can have a really large molecule, a group of molecules. You put it onto a grid shown here. You flash freeze it so that it doesn't fall apart. You use electron microscope to image, and you get this kind of blobby looking thing, which is a map. And then using a whole variety of methods, you get a final model. This has now become the most popular method. It's growing very rapidly for structure determination, and it allows you to look at really, really large samples. So far I've told you about single methods, but in fact, as time has gone on, there are multiple methods that are being used to determine structures, and you have to combine them and use computational, various kinds of algorithms. And Carl and I have been collaborating with Andre Sali, who does this, and where you take data from a lot of different methods and you eventually get a model shown on the lower right. So that's called integrative structure determination. And we began working on this about 10 years ago, and we have a prototype for how you process those data to get to be able to visualize the structure. So, okay, so the PDB data are trustworthy, and we got one of these core trust certificates. And the reason is that there's really careful annotation and validation. So in annotation, we check the incoming data for consistency, the sequence and the coordinates, the data range, cross-field checks, standardized nomenclature. We ensure that the data matches information from other data resources, because there are other data resources that say, look only at sequence. And we have to make sure that everything is coordinated. Then we have to do validation. And we do validation of the model itself. Does the geometry of the model make sense? But we also do the model versus experimental data. And that's why that thing that happened in 2008 saying the experimental data had to be available allowed us to come up with validation methods. So this is just a schematic of the pipeline. We have a single system. It's called OneDep. It's a collaboration of the worldwide PDB, all the different data centers to have this kind of a pipeline. The data comes in, and then the different parts are validated and checked. The ligands, which are the small molecules that sometimes attach to the proteins. For example, in hemoglobin, you have a heme, and we have to check that geometry. Yes.
Who pays for all of this?
NIH, NSF, and DOE. And
DOE.
yep, because we have a lot of structures that are relevant to energy. We have to show relevance for each thing. That my talk isn't to show that, but I could give another talk about all the impact in the different fields. So we have to do sequence processing. We have to compare the sequence that's in the file with known sequences that are in other databases. We do various kinds of other kinds of annotation, and then we do validation, meaning checking the model and checking the model against the map and so on. And then we have various kinds of graphics that allow you to visualize just how bad or good the structure is. And then once this is all done, the data are disseminated. One group called the RCSB that I used to be in charge of is the archive keeper. So there's a master archive, but all the other groups copy that data and mirrors and distribute the same exact data. For the validation, we do the geometric quality, the experimental data quality. I'll show you that in a minute. And what's something called goodness of fit. We have method-specific validation for X-ray, that's MX, EM, and NMR. And then each component is validated, the polymer, the ligand, and the short oligomers. And then this is all given as a report. It's a very lengthy report, which people take very seriously. And when you submit your paper, you have to submit your validation report. So that because otherwise, there's no way, we do not give the coordinates to the reviewers, but they do get the validation report. And they can contact the author for the coordinates, and these days people will do whatever the reviewer asks them to do. And so this is an example of what a slider of a pretty decent structure, and has some of the validation measures that we do. In the geometry, we look at simple things like bond lengths, bond angles, clashes, whether things are too close to one another, whether they're Ramachandran outliers, chirality issues, and side chain conformers. Now, I'll say something after I show you a couple more things. We then also do the map model fitting, and we produce the electron density maps, and then we look residue by residue to see if there's anything that isn't fitting. And so green is good, red is bad, and orange is somewhere in between. And all of this is given back to the author, and if they get it right away, and then they're able to, and they can also do it before deposition, and they can correct errors that they may have made. And then for the ligands, this is an example on the top of a very well fit ligand where everything is an electron density. And then on the bottom is this mishmash of not such a well determined ligand. All of that's checked during the validation process. So how long does that take? It takes from the time of deposition to the time of sort of acceptance and everything being finished is about 2 weeks. The actual process is a few minutes, but there's a lot of back and forth with the authors to make sure everything is in place. It's been the same 2 weeks for 25 years. And the number of staff are now fewer than we used to have because we have more and more automation. So we couldn't have it that just because we have more structures, you have more people. So we had to make very efficient methods for doing all of this checking. The way in which things are checked, is determined by communities. We have a whole series of task forces that were set up with experts in each field and I've been on, now that I'm no longer in charge of the PDB, I'm on some of these task forces and they go on for months and years of fussing about what exactly should we be doing. And then ultimately what happens is we write a white paper which says, okay, this is what you should be checking for X-ray. This is what you should be checking for NMR and so on. And so I think you're beginning to see that there are hundreds or maybe thousands of people involved in making this all work. So today, when we started the PDB, there were 7 structures. Now there's 215,000 structures. We have the worldwide distribution sites. All the different sites, the processing of data is done by region. So the European structures are done in Europe and the Chinese structures are done in China and so on. And but if somebody has an earthquake, like in Japan, everybody will take their structures and process them because none of us can afford to have anything slow down. So that, and the PDB is used by all sorts of people for all sorts of reasons in research and education. And there's a significant number of children who we have various kinds of things to teach children about structure. And the PDB is FAIR, which is a relatively new term. The data are findable, accessible, interoperable, and reusable. On the right, I try to defend or tell you which aspect of the PDB makes it this way. There are actually now committees that review all databases in biology to say whether or not they're fair. And of course, one of them is called the Global Biological Core Resources, and there are a bunch of data resources that are reviewed, have been reviewed, and then the minimum requirement is that they be fair. In terms of usage, there are 17,000 new structure depositions per year. The responsibilities are distributed by geographic location. In 2021, there were 2.3 billion data files. So a data file of all the coordinates downloaded. And one of the groups that downloaded a lot of structures is AlphaFold, and I'll talk to you about that in a minute. The data are used by more than 400 different biological databases, so the reuse is extensive. Okay, so there's another activity which is very fortunate. It's called the Critical Assessment of Methods of Protein Structure Prediction, CASP. It's an organization whose aim is to predict protein structure from sequence. The modeling targets are identified every 2 years. The targets are structures that are about to be solved, but are not yet in the PDB. It's quite complicated to get this because we never want to hold up anything in the PDB, but we need to have some testing going on. And so there are these group of structures that are part of the CASP. competition. So then people in CASP tried to predict structures of these molecules that they don't know what the structure is. PDB knows, but they don't know. And they have to return the results at 3 weeks. The results are then assessed by comparing predictions to solved structures, and then the assessments are done by protein structure experts. In 2020, there were 97 research groups that participated in this, 215 modeling methods. There were 67,000 predictions. There were 52 proteins received. And in that year, there was an extraordinary increase in accuracy of the computed 3D protein structures. And AlphaFold, which is part of DeepMind, which is a Google company, submitted models that were competitive with experimental accuracy. So that was why there was a big deal and a lot of publicity, and it's probably one of the most successful uses of AI. The way it was done, I cannot tell you the methodologies, but I can give you the references. But I do know that it was trained on 100,000 protein data downloaded in 2019. I also saw the activity because we used to have a chart. We could see where there was a particularly large amount of download activity. And this was an electronic chart. And we saw San Francisco, New York, London kept having a huge amount of activity. So I said, these are not structural biologists. I don't know who they are. Well, now we know who they are, but that was very interesting. And I am told by the people in AlphaFold, or in DeepMind, that the high level of curation of PDB data combined with the availability in machine-readable form was key to their success. They also told me that when they used the legacy format, not the MMSIF, they couldn't get anywhere. Because we're dealing with computer engineers who are not going to have to go reading articles to figure out what each column is in an 80-column format. Whereas with SIF, it was very easy for them to figure out what was going on. There's also, and this is a lot of talk, and I think it's true, the incredible resources that they had at their disposal for doing their work, which you would understand better than I understand, was just superior to any of the other groups who were all academic. And in the article describing all of this by the CASP group, they talk about, maybe we're doing this all wrong. And in terms of the academic versus the industrial. But I can't comment on that. I'm just, I'm referring you can look at the article to see. There is an AlphaFold database that's hosted at EBI that has all the models, all the predicted. You could take any structure and get a predicted model at that particular database. The RCSB, which is the American branch of the PDB, has on its website the ability to download models and compare them with the experimental models and the various kinds of presentations of the features of these structures. So AlphaFold for the when AlphaFold came to be and when we got the results, there were many crystallographers who were very, very upset because they said, we're going to be out of business because these guys can do it all. And my view was very different because this was the whole reason we wanted to have a PDB is like make it as simple as possible to get the majority of structures, and that would be great. So I was really happy, and it reminds me of in 1973 when we had the war on cancer began. I was working in a cancer center, And people at the cancer center said, well, what are we going to do? What kind of jobs are we going to have if they cure cancer? And I said, hooray, So that's how I feel about AlphaFold. There's still problems, there's still challenges, there's lots of work going on. It's made it possible, it's opened up the whole field of structural biology in ways that were unimaginable. So simple structures will be predicted pretty damn well by AlphaFold. Complicated things that are big assemblies and things having to do with ligands are still a challenge, and it's just pushing the field forward. So in my personal view, this is a fantastic thing for science. And it also, when I read about what's going on in AI in general, and again, I'm in no way, in any way, an expert, I think if we could get this level of curation in other fields, I think we would have, I think it would be way better. You asked the question, how much does it cost? It costs a lot of money, but you get a good result. And so, and the people who do the work, I mean, we've pushed the field forward even before AlphaFold, just by really making sure the structures are reliable. So that's what
I wasn't implying that I thought it wasn't worth it. I was just wondering,
No,
this
of course,
is a huge community effort.
I'm glad that you see it. Not everybody. I remember the first time a friend of mine from Einstein Medical School came to visit the PDB. He said, "Where's Monica?" I said, "What are you talking about?" He said, "The one who fixes all the data." I said, "There's a team here, and she's one person. Monica, all of the curators are PhD scientists and when they leave, they go to either Google, maybe not now, but they used to go to Google, or they become assistant professors somewhere. I mean, they're trained scientists. But a lot of people think, oh, all you're doing is fixing spelling or something. They don't realize quite how much is done. And they do now, but back in the day. So what next? AlphaFold will accelerate structure determination of simple structures, no doubt about it. That will free time and resources for solving more complex things. And this will lead to a vast acceleration of structural biology and allow us to go to the next frontier. Here I have a slide that is made by our colleague Andre Sali, which shows the evolution or revolution of structural biology, where you have on the left the structure of DNA, which is actually, as Andre has pointed out, the first integrated structure, because it would really, you had to take a lot of information from a lot of different fields to get that double helix. And then we have the structure of myoglobin, which was the very first structure. And then using integrated methods, Andre's group has done this nuclear pore complex, which is a very complex machine. And now that opens the door to looking at things that are really, real next challenge is cell biology, and to be able to understand the human cell is something that it used to be like impossible to even contemplate. And now there are several groups trying to do that. And it is a very complicated thing because, of course, what's in the human cell are all different kinds of molecules and organs, organelles, and so on. And we have to understand their structures, their interactions, their motion. And unlike some of the simple-minded things that are shown in textbooks, there's all these molecules, millions of them floating around everywhere, interacting with one another. And some of us would like to understand that. So the next frontier is cell mapping. And this is a project that Andre and Carl and myself are very involved in. And this particular part of it is creating a cyber infrastructure that will allow us to handle all the data and models and software involved in cell mapping. And so this is a project that we're trying to launch, and I'm very excited about that. And I tried to put just things that I talked about, which is the structural data ecosystem, which has, of course, the PDB, but we have NMR database, EM database, PDB dev, which is for the integrator, EMDR, which has to do with electron microscopy, and then cell lab. So these are all in some way related to one another and key to, in my view, that the kind of framework that we set up with the PDB can just keep extending to more and more complex systems. So that's what I have to say. To do what?
To contribute.
Oh, now that's a very interesting thing. So I didn't talk about this part of things, but every drug, well, 80%, 90% of the drugs that are developed depend on the PDB. Every pharma company downloads the PDB every week or every other week, whatever, the whole thing. They don't use the website at all because they don't want anyone to know what they're doing. Fine. So, but they're also doing the structures themselves. Getting those structures from them has been impossible. And there are like a whole lot of reasons. And it would be great if we could, because the more data we have, so their argument is if they do a crystal structure and then it turns out that it's not the right target, the ligand is binding properly or whatever it is, they don't do what's called refinement. So the structure is low quality and they don't want to give us low quality structures, but they could, and we could, I'm not going to do, but the PDB could refine those structures and get them to the quality. So that's an issue. And that's been an issue with the pharma because they're using the PDB. They don't support it in any way financially. And they don't give us the data, but they use our data. If anyone who understands how to change that, I'd be very happy to hear. AlphaFold, on the other hand, so DeepMind, I've spoken to the people who did the work, and it's very hard to get them to acknowledge. I mean, they acknowledge what we did, but wouldn't it be nice to provide a little bit of support because they totally depended on us. There are now discussions going on with them that look very positive. In terms of, we're not asking for operational support. I don't think that's appropriate, but support for some of the outreach activities would be really fantastic.
So we have two questions from Adam from the audience. So first question, what are your thoughts on AIX biosafety? Are you worried that this kind of advance could lead to really unwanted outcomes in the designing bad things? How should we think about this?
I don't know how to think about it. Tell me, explain it to me, translate that to me.
Adam, are you on?
Part of the question is like what types of
Yeah, now I'm here.
Okay, yeah, Adam.
Yeah, I mean, if AI can accelerate our ability to decode and potentially design a novel protein, you can also do really bad things, right? So how do you think about that?
Sorry.
I think I can translate Adam's question.
Okay.
And he's interested if they could be, you can use these proteins to design like dangerous proteins that you can be risks to human.
Dangerous protein.
Yeah, kind of the risks of having all this data being used for like bad bad outcomes. Like you have
Well, we have,
Oxford
we have
professor.
COVID, we have all the dangerous stuff in there.
Right, but presumably people could design biological weapons. or
Yes, they
things like
could.
that based on that.
Yeah, they
And
could.
Is there any concern, and are you doing any, not you personally, but are you doing anything to monitor that, prevent it?
No. So there's no, nobody can be rejected from the PDB. So if you have a structure, which is either technically not strong, or in some way, we're talking about the ethical thing of some kind of a really bad. So anything, like for example, with COVID, I'll tell you something about what happened there. With COVID, as soon as that sequence was made available, somebody did the structure of some part of COVID. All of the COVID-related structures were put in the PDB, and all of the people who did that did it without publication. They made it immediately available. Same thing happened with HIV. So there was a public health risk. Then the community got together and said, we have to get this out the door because that's the only way we're going to be able to design, antivirals or vaccines or whatever. So whether people take these things and then do something bad with it, there's always that possibility, but it hasn't happened yet as far as I know, but I don't know. But nothing is rejected, so.
So there was a second question. What are the most successful failures of PDB design or deployment? That is, what mistakes were made and that we learned a lot from about how or how not to build these tools?
Yes, okay, the first mistake was to use this, the original PDB was an 80-column format with flat files. It was not in any kind of a proper database at all. In the early days, that was okay. It was not, it became more and more not okay as there were more structures, and there was resistance. So the original PDB was at Brookhaven, and over the years, there got to be a kind of almost no development going on. That was bad, okay? Then when we took it over, now I was at Brookhaven doing that stuff at the beginning, but then nothing advanced. And then when we decided we would do it and we competed for the contract, there were articles written that I was going to be a danger because I had these very forward-looking ideas and that was going to destroy the PDB, and we like it just the way it is. And so I had to live with that for a few years until we demonstrated that it was actually better to do it the way we would do it. But, it takes a long time for people to accept change. And, so I don't know if it was it wasn't an error at the beginning to have this kind of flat format, but it was an error to not move more quickly. That was an error. There's also an error in the modern PDB. There are serious problems with the search capabilities, which I take issue with some of the search mechanisms that are used, and that could be a whole lot better. And maybe we'll make it that well. I can only kibitz on the side now, but I'm hoping that we'll make it better.
I think Adam is also interested in some of your thoughts on kind of the community building aspects.
Community building. I was a natural community builder just by instinct, but I had no idea what I was doing at all, okay? Absolutely no idea. But then I got involved with this group that I highly recommend. It's called the Stakeholder Alliance. It's run by Joel Gershenfeld, and he's a sociologist. And he talks about how you align communities and how you make consortia and how you build these kinds of things like the PDB. And so now I actually understand the principles, okay? And so there's Top down, no good. This bottom up is pretty good as long as the organization doesn't get too big. But then there is this minimum viable consortium, and that idea is that you have these separate groups that operate independently, but then there's crossover between them. And that apparently is what we did with the PDB unknowingly. So we learned, we did it because it seemed like a good idea. And then when I joined Joel's group to study this, I realized that what we had done. So it's this idea that you have, say, PDB Europe, PDB Japan, PDB America, all different the way they run, completely, and nobody controls that. But there are certain things that we agree on. We focus on what we agree on, and we let everybody does what they need to do. And that's human nature. And a sociologist, and there in this group, there are scientists and sociologists and psychologists and all business people. And so I've learned from that. So I learned after the fact. So bottom up is Eleanor Ostrom, who got a Nobel Prize in economics in, it was 2009. And she believed that you do everything bottom up. And I think at the very beginning of the CDB was bottom up.
Any other questions? Anything else from the? Okay, well, with that, I think we're at time. Thank you very much. It was great.